So we'll just wait for 5 minutes and then we'll start the session, okay? Is my voice audible? Am I audible to everybody? I hope my voice is audible. We'll wait for 2 more minutes and then we'll start the session, okay? Okay, so I'm starting the class now. This is week. Fine. So I hope my voice is audible to everybody. So good evening, everyone. Today we are going to discuss the questions from week 2 lecture. So week 2 lecture is basically about differential gene expression, and we will be dealing the questions of related to differential gene expression topic, and simultaneously we will try to discuss the concepts which are related to this topic. So in the last class we have completed week 1, and now we are going to see the week 2 lecture questions. Just a minute. Okay. So before we start the questions, there is a concept which is known as genome equivalence. Now, what is the meaning of genome equivalence? The concept, excuse me, the concept that I'm talking is genome equivalence. Okay. So, what is the meaning of genome equivalence? Genome equivalence is a concept which says that all the somatic cells in an organism contain the same DNA. That means same set of genes are present in all the somatic cells of an organism. Regardless of the function of the cell, regardless of the level of differentiation or the location of the cell, all the cells of the body have the same DNA, that is the same set of genes. That means every cell has the complete genetic blueprint of the entire genes which are present in it in the organism. Okay, so that means what does this concept says? That all the somatic cells, all the somatic cells in an organism contain the same set of genes, that means the same DNA, contains the same DNA, that is the same set of genes. So now if we give this information, this means that all the DNA of a cell which is present in eye and the DNA of a cell which is present in liver are the same. Okay, same set of genes are present in eye and same set of genes are present in the liver. But how come the function of liver and eye cells are different? How is it possible? There are genes of eye color which are also present in liver cell, and there are genes for liver cells which are also present in eye cells. Then why liver cells does not exhibit the function of eyes and eye cells does not exhibit the function of liver? Can anybody answer the question? Are you able to follow what I'm trying to say? That if there is the concept of genome equivalence, which means that all the somatic cells of an organism have the same genetic composition, same genetic makeup, then how come the differential expression of genes happen? How come not all the genes are expressed? The phenomena that governs this is differential gene expression. What does it mean? It means that although all the genes are present in all the cells of the body, not all the genes are expressed in all the cells of the body. And not all the genes are expressed at the very same time. This means that although liver genes are present in eye cells and eye genes are present in liver cells, but eye genes are not expressed in the liver cells and liver genes are not expressed in the eye cells. That means that cells or the genetic makeup, it exhibit the property of what we call it as differential gene expression. Okay, differential gene expression. And how this differential gene expression occurs? What are the factors that regulates this differential gene expression, and how this differential gene expression ultimately helps in the proper functioning of an organism, that we'll study. I hope this much is clear to everybody. This was just to give you a brief background of what we are going to deal today in today's class. First, we understood about the concept of genome equivalence. That then we saw that although there is genome equivalence, but not all the genes are expressed, because there is a phenomena which is called as, there is a concept which is called as differential gene expression that occurs in our body. Fine. Now let us come to the first question, which is, which of the following are the reasons why the Briggs and King experiment was insufficient to demonstrate the genomic equivalence? Now you know what is the concept of genomic equivalence. The nucleus was not from a differentiated somatic cell. The nucleus was unipotent. The experiment did not progress past the gastrula stage, or the experiment did not progress past the blastula stage. This is the question. I will give you 2 minutes. Try to solve, and after whatever answer you think is correct, just write it in the message box. And after 2 minutes, I will switch the screen and we will discuss the question. Okay. So right now it is 7:09. I will switch the screen at 7:11. So try to solve this question. Think. One more minute left. Okay, I'm switching the screen now. It's 7:11. Let's see whether you have answered something or not. Nobody has answered anything. Why? You couldn't understand the question? Okay, fine. Let me just see. So the question is, which of the following are the reasons why the Briggs and King's experiment was insufficient to demonstrate the genomic equivalence? The nucleus was not from a differentiated somatic cell. The nucleus was unipotent. The experiment did not progress past the gastrula stage, or the experiment did not progress past the blastula stage. Okay. Now, let us try to understand this question. So, what does the Briggs and King experiment suggest? So basically, what they did was that they transplanted the nuclei from early frog blastula cells into an enucleated eggs to see if those nuclei could support the full development. Okay, so the correct answer would be the nucleus was not from a differentiated somatic cell. This is the correct answer, and the experiment did not progress past the gastrula stage. Let me see. Yes. So see how they are the correct option. First thing is what they did was that they transplanted nuclei from early frog blastula cells. That means they have taken the nuclei from early cells, early blastula cells. That means these were not differentiated cells. So here it is written the nucleus was not from a differentiated somatic cells. This is the correct statement because they have taken early blastula stage. Okay, they have taken early blastula stage to take the nucleus. So early blastula stage means it is an undifferentiated state, obviously. So the first option is correct. The nucleus was not from an undifferentiated from a differentiated somatic cell because they have used early embryonic cells which are not differentiated somatic cells. Therefore, the experiment didn't fully prove the genomic equivalence, which requires showing a differentiated nucleus still contains the full genome. Okay. Then next is the experiment did not progress the gastrula stage. Why this is the correct answer? Because in many cases, the nuclei are taken from the later stage embryo, such as gastrula or pleurula. What happens? the nuclear transfer is incomplete or it basically stops. So what happens? The the organism or the embryo dies. So we cannot reach the later stages. So therefore, these are the two correct statements. Why second option is incorrect? Second option is the nucleus was unipotent. The transplanted nuclei, it actually came from a multipotent cells, not unipotent cells. So this is wrong. Then the experiment did not progress past the blastula stage. No, they passed the blastula stage, but after blastula stage, they couldn't reach the gastrula stage. So therefore, these two are the correct statements. Okay. Fine. Now the next question is, which was shown what was shown with Boveri's experiment using an Ascaris embryo? First question is, the X cytoplasm is the main driver of embryogenesis. The number of sperm nuclei entering the egg is irrelevant to embryogenesis. The number of chromosomes is vital for embryogenesis. And the fourth one is, the sperm pronuclei can divide multiple times before fusing with the egg pronucleus. Okay, so you have to give the answer that using the Boveri's experiment uh, with the help of Ascaris embryo, what was shown? These are the options. I'll give you 2 minutes to answer the question and then I'll switch the screen and then we'll discuss the question. Okay, right now it is 7:16. I will switch the screen at 7:18 approximately. Try to solve this question. See all the options and then try to understand what they are asking. Okay, try to answer this. I'll switch the screen in a few seconds. Fine, I'm switching the screen now. Whether you have answered. Okay, so Wahida has answered third. Third is, the number of chromosomes is vital for embryogenesis. And yes, this is the correct answer. Good, Wahida, you have answered it very correct. So the question is, with what was shown with Boveri's experiment using an Ascaris embryo. So what is this Boveri's experiment? Boveri's has conducted experiment with Ascaris and also with sea urchin. Okay, so Boveri's experiment has demonstrated the role of chromosomes in development. Okay, so it has demonstrated the role of chromosomes in development. So what he did was that he manipulated the fertilization process in such a way that embryos were created with abnormal number of chromosomes. Okay, so there were abnormal number of chromosomes in the embryo. How could he create a normal number of chromosomes in the embryo? Because he has used multiple sperm to fertilize one egg. So he has used polyspermy. Okay. Polyspermy means that he has used multiple sperms to fertilize one egg, and this lead to cells with extra chromosomes or missing chromosomes. Then what he observed, that embryos with the wrong number of chromosomes, they fail to develop normally, whereas embryos with correct number of chromosomes, they develop normally. This means that the number of chromosomes is vital for embryogenesis. Okay, so this is what he showed with Ascaris embryo, that number of chromosomes are vital for embryogenesis, because when he created some kinds of embryo which has abnormal number of chromosomes, that means more extra chromosomes or lesser chromosome than the normal level, then those embryos fail to develop normally, whereas the embryos which has the correct chromosome, they develop normally. So what is the conclusion? Each chromosome has a unique role, and the correct number of chromosome is essential for the proper embryogenesis process. And this was actually one of the earliest experiments that has actually shown that chromosomes have the genes, or chromosome have the instructions that actually determines how an organism will be formed, something that now we call as genes. Okay, so therefore option third is the correct one. Now let us see what is the first option. The first option is the egg cytoplasm is the main driver of embryogenesis. Was this the main gist of the theory? No, this is basically more about the cytoplasmic determinants, which is not his theory. This is someone else's theory. It is basically Spearman's theory of cytoplasmic inheritance. Then the number of sperm nuclei entering the egg is irrelevant to embryogenesis. No, actually, the number of sperm nuclei which are entering the cell is very much relevant, because that only will decide that how many nuclei are actually going inside the cell, and whether it will have correct number of chromosomes or not. So this is actually completely the opposite of what he actually told. Third is the correct option. Fourth is the sperm pronuclei can divide multiple times before fusing the egg nucleus. This was not the focus of Boveri's experiment. Boveri's experiment has shown the importance of chromosomes, which is during embryogenesis specifically. Then the next question is, which of the following experiments seem to support the germplasm theory? First is Boveri's experiment with the sea urchin, Wilson and Stevens experiment with the Drosophila, Boveri's experiment with the Ascaris, yeah, Groudon's experiment with the frog nuclei. So there are four options. So you have to tell that amongst the four, which experiment has supported the germplasm theory. Okay. So right now it is 7:23. I will switch the screen after 2 minutes and then we'll see. Okay. Try to answer this question. Think of all the options. One more minute left. Think what can be the correct answer. Okay, now I'm switching the screen. So Wahida has answered first. That is Bovary's experiment with sea urchin, and this is okay. The correct answer is Bovary's experiment with ascaris. So let us see. See, Bovary's has done experiment with sea urchin and ascaris both. Okay. Now the question here is, which of the following experiments seems to support the germplasm theory? So the germplasm theory was proposed by whom? It was proposed by August Weismann.
Others basement.
Yes, correct. And what does the germplasm theory states? The germplasm theory states that only the germ cells. What are the germ cells? Germ cells are your eggs and your sperm. So only the germ cells can transmit the genetic information from one generation to the next generation. Somatic cells do not influence this hereditary transmission. So the transfer of genetic material from one generation to the other generation happens because of the germ cells. So, according to the theory of germplasm, information flows only from germ cells to somatic cells, not from the somatic cells to germ cells. Now, Bovary's has done experiment with ascaris also, and Bovary's has done experiment with sea urchin also. So, this one, the question that we have just discussed. Okay. So, the experiment that Bovary's did with yeah, Ascaris was that Bovary studied the early embryonic development in Ascaris. Ascaris is what? Ascaris is a parasitic dung beetle. Okay. So he observed that chromosomes were reduced or eliminated in somatic cell lineage, but they retained in the germline cells. Okay. That means the chromosomes, with each round of division in the somatic cells, the chromosomes were getting reduced or eliminated. But in the germline cells, he observed that the chromosomes remains constant. This provided supporting evidence for the Weismann theory that only germ cells retain the full hereditary material, and therefore the Bovary's experiment with Ascaris is the one that supports the germplasm theory. Okay. Wilson and Stevens, what did they do? They identified the role of chromosomal sex determination in Drosophila, which is not related to the germplasm theory. Goodon's experiment with frog nuclei, they demonstrated genomic equivalence. Okay, so this is related to genomic equivalence. This is related to the genomic equivalence. This is related to sex determination. Here, with sea urchin, the focus was more on the genomic content and not the germplasm. Okay, focus was more on genomic content and not the germplasm. But the Boveri's theory of Ascaris, it showed that chromosomes were retained in the germ cells, which supports the germplasm theory. Fine. Then the next question is, which histones are not present in the nucleosome complex? H2A, H2B, H3, and H1. Hmm. It's very easy. Which histones are not present in the nucleosome complex? H2A, H2B, H3, H1. Right now it is 7:31. I'll switch the screen after 2 minutes. Today I'll give you one question as homework. You try to solve that question, and that question we'll discuss in the next class. That will be your homework question. So I'll give you. So what I'm thinking is that I'll be from now on, I'll give you one question as your homework that you can solve, that you can try to solve. Okay. And then that question we can discuss in the next class so that the continuity is also maintained and you show up for the next class also. Fine. Uh, you have 1 more minute left for this question to answer, and then I'll switch the screen. See, okay, now I'm switching the screen. So Wahida has answered fourth option, which is H1, and it is correct answer. And there is one more correct answer, which is H3A. Why? See, H1, first of all, the question is which histones are not present in the nucleosome complex. So, what are the histones that are actually present in the nucleosome complex? It is
H2A, H2B, H3, and H4 are present.
H2B, H3, and
H4,
so
so
is
H1,
there any histone which is named as H3A? No, there is no histone named H3A. You have H2, H2A, H2B, H3, H4.
H4, H3.
And what is H1?
H1 not a histone, it is linker histone.
It's a linker histone. That means
Very
it
good
connects
options.
the two nucleosomes complex.
Associate with DNA.
Yes. So it connects the two nucleosome complex. So the correct answer would be H3A and H1, because H3A there is no histone named H3A. They are H2A, H2B, H3, and H4, which is present in the nucleosome complex, and H1 is a linker histone, which is not present exactly in the nucleosome complex. It is present in between the two nucleosome complex. If this is your nucleosome complex 1 and this is your nucleosome complex 2, there is one histone which is present here, which is a linker histone, which actually links the two nucleosomes, and that is your H1. So it is not exactly present in the nucleosome complex. Fine. Next question is, which of the following histone modifications cannot cause transcriptional activation? Methylation, demethylation, acetylation, or farnesylation? I repeat the question. Which of the following histone modifications cannot cause transcriptional activation? Methylation, demethylation, acetylation, or farnesylation? Okay, think. I'm giving you 2 minutes. Then I will switch the screen. Try to answer this question.
Animation.
1 more minute left. Try to answer this question. Okay, so I am switching the screen now. Let us see what you have answered. Okay. Hmm. So Wahida has answered fourth. I would appreciate if others also participate in answering the questions. It doesn't matter whether you are answering it correct or not. What matters is you are attempting and you are trying to get involved in the class. That's what really matters. Okay? Yes. So Ningaraj has answered acetylation. Ningaraj. So I'm sorry if I'm pronouncing your name incorrectly. Okay. So we have two options now. Fourth one, which is answered by Vahida, and third one. So third and fourth, we have two options. So now let us try to decode the question. Which of the following histone modifications okay, cannot cause transcriptional activation? So to understand this question, first you should know that what histone modifications actually do. So histone modifications, what they do is that they affect the chromatin structure. And when they affect the chromatin structure, it ultimately leads to the control of transcription. That means it can activate the transcription and it can repress the transcription. So in what way you think that it can control the chromatin structure? It can make the chromatin more tightly packed so that there is no space for the transcription factor to come and bind and to start the transcription process. In that case, it is repressing the transcription process. Or in other case, what it can do, it can make the chromatin structure less compact so that now there is ample of space for the transcription factors to come and bind and start the transcription process. So that means these things it can do. So basically, what they do is that the lysine residues, okay, the lysine residues which are present in the histones, they are modified, and the modification in those lysine residues can lead to major changes in transcription. Now, the first one is methylation. Now, methylation generally it represses the transcription, but it can activate the transcription as well, depending on which lysine or arginine residues are getting modified. For example, there is one methylation which is known as H3K4Me3. How we can read it? H3K4Me3, H3K4 trimethylation. We call it as H3K4 trimethylation. And how to read it? H3 means histone 3, K4 means lysine, fourth lysine of the H3 histone is getting trimethylated. This methylation, it activates the transcription. Whereas if there is a modification H3K27Me3, it will repress the transcription. Here, the 27th lysine residue of the H3 histone is getting 3 methylation residues. Here, the 4th lysine residue of the H3 histone has 3 methyl residues attached. So this is the modification. Methylation is the modification. 3 methyl groups are getting attached to the 4th lysine of H3 histone. 3 methyl groups are getting attached to the 27th lysine of H3 histone. So methylation cannot be the correct answer because it can activate transcription also, it can repress transcription also. Demethylation always activate transcription. Okay, so because when you remove the methyl mark, for example, this methyl mark is when removed, then it always activate transcription. Methylation generally it represses transcription. Demethylation generally it activates transcription. But there are some methylation, just like we discussed this one, it activates transcription also. Acetylation always activates transcription because what happens that when you add the acetyl group to the lysine residues, it will neutralize the positive charges. Therefore, the histone and DNA interactions are loosened up. And when the histone and DNA interactions are loosened up, the chromosome becomes much less condensed. So it causes this is the opening of the chromatin. So acetylation can lead to the opening of the chromatin, opening of the chromatin, and therefore acetylation activates transcription. Now we come to farnesylation. What do you think farnesylation will do? Farnesylation is not a histone modification at all. What is it? It is a lipid modification. The attachment of farnesyl group is usually seen in cytoplasmic proteins. For example, RAS protein, the membrane associated proteins. So it is not involved in chromatin regulation or transcription activation or transcription regulation, anything in that sort, because it is not a histone modification. Farnesylation is a lipid modification. So the question was, which of the following histone modification cannot cause transcription activation? This is not a histone modification at all. Okay, so this is the answer. Had it been only these three options with you, methylation, demethylation, or acetylation, in that case, you should have selected methylation, because acetylation and demethylation in all the cases causes transcription activation, whereas methylation can cause activation and cannot cause activation. Generally, it represses. So in that case, you should have chosen methylation. So just like I told you earlier also, which option, it is comparative. The correction of the options is comparative. Okay. Now, the next question is, which of the following does not act as an RNA polymerase binding sequence in a promoter? The question is, which of the following does not act as an RNA polymerase binding sequence in promoter? Tata box, Primno box, Kozak sequence, or B recognition element? Which sequence does not uh, binds the RNA polymerase in the promoter region? That is your question. I'm giving you 2 minutes, and then after 2 minutes, I'll switch the screen. Think. You have 1 more minute left. Okay, now I'm switching the screen. Let us see what you have answered. Okay. So, Gyan Sundari has answered third option, which is the Kozak sequence, and Vahida has also answered Kozak sequence. Okay, fine. And the correct answer is Kozak sequence. So let us try to understand this question. The question is, which of the following does not act as an RNA polymerase binding sequence in a promoter? Excuse me. Now, first is Tata box. Tata box, so we all know it is present in the eukaryotic promoter at around 25 to 30 base pairs upstream of the transcription start site. So it is present in promoter. So this is not the correct answer. Pribna box. Pribna box is present in eukaryotic promoters. So this is also not the correct answer. Then recognition element, B recognition elements. What are these? These are present adjacent to the Tata box and they are also recognized by RNA polymerase. Now comes the Kozak sequence. Kozak sequence is present in the eukaryotic mRNAs, but it is not a part of the promoter region. Okay. Kozak sequence is not a part of the promoter region, first thing, and it plays a role in translation. All these, this one, this one, they all are involved in transcription. But Kozak, it is involved in translation ka initiation. Okay, that means it has no role with RNA polymerase. It does not bind to RNA polymerase. It has no role in transcription process. Therefore, the correct answer is Kozak sequence. Hmm. Next question is, which of the following is a pioneer transcription factor? Myelin, PBX, MEF3, and MEF2. Myelin, PBX, MEF3, and MEF2. Which of the following is an example of a pioneer transcription factor? Right now it is 7:52. I will switch the screen after 2 minutes. To understand this question, you should know first what is the meaning of pioneer transcription factor. Okay. So you do it. We'll discuss after 2 minutes. Okay. 1 more minute left and then I will switch the screen. Fine, I'm switching the screen now. Let us see what you have answered. Okay, so nobody has answered anything. No, you don't know the answer? Fine. So, the question is, which of the following is an example of a pioneer transcription factor? A is MyoD, B PBX, 3 is MEF3, and 4th is MEF2. The correct answer is PBX. Now, what is the meaning of a pioneer transcription factor? A pioneer transcription factor is a factor that binds for the first time to the chromatin. Pioneer means the starting, the initiator. So, since it is binding, it is the initiator of transcription, or it is the initiator factor which binds to the chromatin. It means that it is actually binding to a compacted chromatin. The pioneer transcription factor actually binds to a compacted or a closed chromatin. And after its binding, the chromatin structure actually gets opens up, making accessible for the other transcription factors to come and bind. You understood that the pioneer transcription is a transcription factor which binds to the compacted chromatin. It binds to the compacted, that is closed chromatin, and it actually opens up the chromatin structure. This pioneer transcription factor, after its binding, it opens up the chromatin structure and making it accessible for the other transcription factors to come and bind. Okay, now let us see MyoD. What is MyoD? It is the master regulator of muscle differentiation. And it is not a pioneer transcription factor. It cannot bind to a closed chromatic. So this is not the correct answer. PBX is pre-B cell leukemia homobox. Pre-B cell, pre-B cell leukemia homeobox. Okay, this is PBX. It works with other transcription factors like HOX and MIS, and it can bind to the chromatin which is compacted in form, and it can recruit the other factors, and it has shown to have the pioneer activity. Therefore, this is a correct answer. Then MEF3 is actually a binding site. It is not a transcription factor. This is out of question. The next is MEF2. What is MEF2? It is myocyte enhancer factor 2. Myocyte enhancer factor 2, which basically regulates the muscle and neural gene expression, and it requires accessible chromatin to bind. Therefore, it cannot be a pioneer transcription factor. So the correct answer is PBX1. Okay. Next question is, first we will do this question. Which of the following sites in the genome is often methylated? CPC site, GPC site, CPA site, or CPG site? Hmm. Right now it is 7:59. I will switch the screen after 2 minutes. Think what can be the answer? Okay, now I'm switching the screen. So Wahida has answered fourth, which is CPG, which is actually the correct answer. CPG is the correct answer. So methylation basically occurs in the cytosine followed by the guanine residues. Okay, so CPG islands are the areas where the methylation occurs. So these CPG islands are basically found in the promoter regions, and it is basically present. CPG islands are mostly present in the promoter regions. Okay, promoter regions, and the methylation of CPG islands, it is associated with gene silencing, chromatin condensation, X chromosome inactivation, and genomic imprinting. So these are the factors which helps in CPG islands. Okay, now next question is, this is the last question, and then I'll give you one question as the homework. Okay, this is a big question, so I'll give you 5 minutes for you to understand the question and think. Question is, MECP2 protein is an important reader of DNA methylation. Okay, MEC2 protein is a reader of DNA methylation. Its methyl CPG binding domain, that is the MDB row domain, it recognizes and bind to the 5-methyl cytosine regions. MECP2 is an X-linked and subjected to X inactivation. The MECP2 protein is likely to be involved in repressing several other genes. Which of the following actions is MECP2 likely to perform when recruited to a section of the genome? First is, removes acetylation by recruiting a histone deacetylase, removes methylation by recruiting a histone methyl transferase. Third option is, adds methylation by recruiting a histone methyl transferase. And the last and the fourth option is, adds acetylation by recruiting a histone acetylase. Try to understand this question. This question can be a little confusing. See, it's a big question. Read it, try to understand it, and then we'll solve it. Okay? Read it 2 times, 3 times, as much time you want to take to understand this question. Read it. I'm giving you 3 to 4 minutes to solve this question. Okay? Take your time, understand, and then we'll get back. Read the question, what it is trying to say. The last line is, which of the following actions is MECP2 likely to perform when recruited to a section of the genome? That means when you are recruiting MECP to a genome section, what it will do? Shall I switch the screen now? Okay, I'm switching the screen now. Let us see if you could understand the question. Okay, now let us try to understand this question. The question is saying MECP2 is a MECP2 protein is an important reader of DNA methylation. Okay, it is an important reader of DNA methylation. That means it can read or recognize DNA methylation. First point that we can get from here is that it can recognize uh, the methyl groups. This is the first thing that we can get from here. Its methyl CpG binding domain recognizes and binds 5-methyl cytosine region. That means it can recognize the sorry, which means it can recognize the methyl group. This is what we can understand from here. Okay, just a minute. Huh. So it can recognize and bind. It can recognize the methyl. Then MECP2 is X-linked and subject to X-linked activation. Fine. The MECP2 protein is likely to be involved in repressing several other genes. Now, MECP2 is repressing several other genes. That means in some way it is promoting methylation or removing acetylation. As it is repressing, so it is promoting methylation or activating or repressing acetylation. Then which of the following actions is MECP2 likely to perform when recruited to a section of genome? So now you see the correct answers are it removes acetylation by recruiting a histone deacetylase. This is the correct answer. Removes acetylation by recruiting a histone deacetylase or adds methylation by recruiting methyl transferase. Adds methylation by recruiting a histone methyl transferase. See, it recognizes the methyl group and it represses. That means what happens? It recognizes the methyl group. So if it recognizes the methyl group, it can add the methyl group because it can recognize it. And when it adds the methyl group, it will cause the repression of transcription. And it can, because it is causing repression of transcription, that means what it can do is it can remove the acetyl group also, because removing of the acetyl group or adding of the methyl group causes repression of transcription. So this is important. Addition of methyl group or removal of uh, acetyl group, removal of acetyl group, it causes repression of transcription. So, so removal of acetyl group will cause repression of transcription. Addition of methyl group will cause repression of transcription. Removes methylation by recruiting a histone methyl transferase. No, because if you are removing the methylation, it will not cause repression, it will cause activation. Adds acetylation by recruiting a histone acetylation. No, because if you are adding acetylation, what will happen? If you are adding the acetyl group, it will cause the activation of transcription. So it is a very easy question. If you see, only the thing that you have to remember here is that how the question is framed. The question looks so big. It's such a big question. It looks like it's a very big question. But if you try to understand, and when you understand it, so you find that it's very easy. Fine. Now let us come to the next question, which is actually a homework for you guys. The question is, if a woman heterozygous for deletion in the Prader-Willi syndrome locus had a child with a man heterozygous for deletion in the Angelman syndrome locus, what is the probability for this child to suffer the symptoms of Prader-Willi syndrome? Okay, I'm repeating the question. The question is, if a woman heterozygous for deletion in the Prader-Willi syndrome locus had a child with a man heterozygous for deletion in the Angelman syndrome locus, what is the probability for this child to suffer the symptoms of Prader-Willi syndrome? And the correct answer is 1 by 4. I'm telling you the answer priorly. This is 1 by 4 is the correct answer. Okay, so you have to solve this question. So just quick hints to solve this question are, Prader-Willi syndrome. So Prader-Willi syndrome basically is a paternal deletion. Okay, Prader-Willi syndrome is a paternal deletion, and Angelman syndrome is a maternal deletion. In both the syndrome, the other allele is normally silenced via genomic imprinting. So a deletion from the wrong parent leads to disease. Okay, I'm just writing it over here. Prader Willi syndrome. It's a paternal deletion. And Angelman syndrome. Angelman It's a paternal deletion. Okay. So what happens? Hmm. In both the syndromes, the other allele is normally silenced. Okay. So paternal genotype में what will happen? Heterozygous mother will be heterozygous for deletion and deletion will be on the maternal chromosome. Whereas father will be heterozygous for the deletion in the AS locus, whatever the locus is there, and deletion will be on the paternal chromosome. So what does it mean? It means the mother has one normal paternal gene and one deleted maternal gene, whereas the father has one normal maternal gene and one deleted paternal gene. Okay, and child gets one chromosome 15 from each parent. So paternal chromosome is deleted and mother chromosome, maternal chromosome is imprinted, that is inactive. Okay, so I have given you probably all the hints that are needed to solve this question. And it would be very good if you could solve this question and come back in the next class. So in today's class, we have started with the genome equivalence. What is the meaning of genome equivalence? And although there is a phenomena which is known as genome equivalence, then also how this genomic imprinting comes into picture. Okay, that was our this thing. And then we studied the various theories and what were the various theories. Let us to quickly try to see. Hmm. We start with Briggs and King's experiments, and then we studied about the Boveri's experiments. So Boveri has conducted experiment in Ascaris and in sea urchin both. And then we started with the August Weismann germplasm theory and how the Boveri's experiment has actually supported the germplasm theory. And then we went to the differential gene expression. Okay, and then we saw after differential gene expression we saw the different modifications, methylation and demethylation, acetylation, farnesylation, how they are important and how they causes the histone modifications, which helps in whether the activation of transcription or repression of transcription. And then we solved some few more questions. What are the pioneer transcription factors? Just a minute. Huh. So then we studied about the pioneer transcription factor. What is the meaning of pioneer transcription factor? So pioneer transcription factor, the transcription factors which bind to the compact chromatin, that is a closed chromatin, and the PBX, which is your pre-B cell leukemia homeobox, it is a pioneer transcription factors. So basically, pioneer transcription factor, it binds to the compact chromatin and then it opens up the chromatin structure. Okay, and then we have studied about this very big question, which actually looked big, but it was just conceptual question which needed to be solved and dealt logically. And then we solved this question about the methylation, and this is the question that has been given to you as a homework, although we have already discussed the question here itself in much detail. Okay, so just a minute. So do you guys have any questions? If you have any questions, please let me know. Do you have any questions? Okay, so then I think just a minute. Mm. So I think if there are no questions, we can just end the class. Okay. So, okay. We'll meet in the next class. Bye, everybody. Just a minute. What's happening?